In medicine, confusion costs marks and mistakes cost lives. Stop memorizing, start understanding. This is Dr. ABM, where medicine finally makes sense. And today, we'll be looking into cell biochemistry, everything you need to know about cell biochemistry. So let's get right into it. Now, a cell is the smallest, simplest basic structure and functional unit of life. It can exist on its own and carry out essential functions of life. Now, the cell theory was propounded by scientists Matthias Sledin and Theodor Schwann in 1839, and the cell theory is made up of the following things. Number 1, all living things are made up of one or more cells. 2, cell is the basic structure and functional unit of life. 3, all living things are either unicellular or multicellular. 4, all existing cells are formed from the division of pre-existing cells. And 5, cells contain hereditary information which is passed down from one generation to another. Now, having discussed the concept of what a cell is, it's important you know about the organization. Now, a cell is made up of components. These components are referred to as cell organelles. So, the cell organelles are the lysosome, the ribosome, the cell membrane, the smooth endoplasmic reticulum and the rough endoplasmic reticulum, the nucleolus, the nucleus in general, the cytoplasm, the Golgi apparatus, the peroxisome, the centrosome, centriole, the lysosome, the mitochondria. Alright? So these are the cellular organelles, and it's important we discuss their function. Now we'll start with what the cell membrane is. Now, the cell membrane is a phospholipid bilayer that controls what goes in and comes out of the cell. Now, the theory that explains the cell membrane is the fluid mosaic theory. The fluid mosaic theory says that the cell membrane is a phospholipid bilayer with an hydrophilic head and 2 hydrophobic tails. Again, the cell membrane is a phospholipid bilayer with an hydrophilic head and 2 hydrophobic tails. The hydrophilic head are water-loving and they point outwards, while the hydrophobic tail are water-eating and they point inward. It's important you know that anything that is hydrophilic is lipophobic, and anything that is hydrophobic is lipophilic. And that's the cell membrane. Now, the cell membrane is made up of components, usually 3 things: lipids, of which the most abundant is phospholipids; proteins, which are 2. You have integral proteins and you have peripheral proteins. The integral proteins are found deep within the cell, lie inside, and they act as transmembrane channels, while the peripheral proteins are usually on the surfaces of the cell membrane. And then you have carbohydrates. Now, the carbohydrates that you have there are in conjugated form, so we call them glycocalyx, and they can be glycoproteins, glycolipids, usually in conjugated form. That's the cell membrane. Now, it is the cell membrane that regulates what goes in and out of the cell, and hence we talk about transport across the cell membrane. But before then, let's quickly discuss all the other cellular organelles and note their function. Now, we'll start with the cytoplasm. The cytoplasm is the space that anchors all cellular organelles except the nucleus. So this space, entire space inside the cell, is referred to as the cytoplasm. It contains a fluid known as cytosol and the site of chemical reaction. So that's the cytoplasm. And then moving on, it's a very important structure. The mitochondria is the powerhouse of the cell. It's a site of oxidative phosphorylation and ATP generation. Now, the mitochondria has a double-layered membrane. It has an outer membrane and it has an inner membrane. This is the outer one and this is the inner one. Right? So it has a double membrane. There are 2 organelles in the cell that have a double membrane, the mitochondria and the nucleus. Now, the inner membrane is folded. You can see folding structures. Right? This fold structure is regarded as a cristae. It's very important. It's regarded as a cristae. So the mitochondria has its own DNA. Is called non-chromosomal DNA and is a specific feature of the mitochondria. So the mitochondria is the powerhouse, hence is the site of generation of energy and ATP. Take note. So because the mitochondria can correct and control its own activities, the mitochondria is regarded as a mini cell. It's very important. The mitochondria is known as a mini cell. Take note. Alright, so Other organelles that we have is the Golgi apparatus. This is the Golgi apparatus. The Golgi apparatus is responsible for packaging and repackaging of proteins. Take note, and it is made up of a cysteine, a folding. Then you have the peroxisomes. The peroxisomes are responsible for degrading and breaking down peroxides and fatty acids. Then you have the endoplasmic reticulum. Now the endoplasmic reticulum are two types. There's the smooth and there's the rough. Now you see the surface of this rough, it's something is attached to it. Those things on the surfaces are ribosomes, and then the smooth does not have it. So the rough endoplasmic reticulum plays a role in protein synthesis because it has ribosomes, and the smooth endoplasmic reticulum is for cholesterol and lipid synthesis. And then you have the ribosomes. The ribosomes are the site of protein generation and synthesis in the cell. They contain RNA known as ribosomal RNA. Check out videos where RNA is discussed in details. And then you have the centrioles, where they occur in pairs. They are called centrosome. The centrioles are responsible for formation of spindle fibers and basal organelles, basal cells. Then the centrioles are responsible for formation of locomotive structure in cellular organisms such as cilia, flagella, pseudopodia. It's very important. Then you have the lysosome. The lysosome are regarded as the garbage system of the cell. It's also regarded as a suicide bag. It contains hydrolytic enzymes that break down structures. So the lysosome is responsible for degrading worn out structures or worn out cells. Hence the reason why it's called suicide bag. So these are structures you have within the cell. And then you have the nucleus, right? The nucleus is made up of a central portion called the nucleoplasm, and then you have a dense region called the nucleolus. Now inside the nucleoplasm there's a nucleus, or a fluid-like, semi-solid fluid-like structure. Then you have the nucleolus. The nucleolus is the site of RNA synthesis, is a dark staining zone. Now the nucleus is regarded as the brain of the cell, and that's because it contains chromosomal DNA. DNA is the oxyribonucleic acid that is responsible for controlling all the activities within the cell. So it is the nucleus that produces proteins that direct all the things that occur within the cell. Now these are structures that you should know about the cell membrane. We have discussed the plasma membrane, which is the cell membrane, and these are other structures you should know about the cell. Now moving on, we begin to talk about the cell membrane and transport within the cell membrane. Now moving on, we'll talk about membrane transport. Now transport through the cell is regulated by the cell membrane, and the cell membrane is a phospholipid bilayer. Hence it allows organic substances that are lipophilic and hydrophobic to easily pass, while inorganic substances that are hydrophilic and lipophobic do not easily pass. Hence there's a mechanism of transport through the cell membrane. Now there are three types of transport you should know. There's passive transport, there's active transport, and there's vesicular. We'll start with passive. Now passive means, number one, energy is not involved. Number two, it is movement from high concentration to low concentration. So it's a downhill transport. And number three, carrier proteins are not involved except for facilitated diffusion. So these are the three conditions for passive transport. Now what are the examples or types of passive transport? The first one we have is simple diffusion. So diffusion is the process of or the movement of molecules from a region of high concentration to a region of low concentration until a state of equilibrium is attained. An example of diffusion in the body occurs in the lungs, where we diffuse oxygen in and we diffuse carbon dioxide out, right? So that's an example of simple diffusion. Remember, all of these are under passive transport. Another example of passive transport you have is facilitated diffusion. This is a type of passive transport where movement is occurring from high concentration to low concentration with the aid of a specific transporter or transport protein. An example of facilitated diffusion is transport of glucose via the glucose transporters. Take note. Another example of passive transport is osmosis. Osmosis is movement of water molecules. Water molecules, we move from where water is much to where water is low. So it's moving from a region of high water to a region of low water. But take note that means region is moving from a region of low concentration, because concentration is defined in terms of the solute. So moving from low concentration of solute to high concentration of solute, but moving from high region of water to low region of water. So osmosis occurs through semi-permeable membrane all over the body. So these are examples of passive transport. Moving on, we move to active transport. Now, active transport occurs under the following conditions. Number one, it involves carrier proteins. In this case, we are using carrier proteins. Please take note. Number two, it is an uphill transport. You are moving from low to high. So the poor is giving the richer, or the rich. The poor is giving the rich, hence why it is involving energy. And the third one is that energy is involved. So the three condition is that it involves carrier proteins or transport proteins, is moving from low to high concentration, and energy is involved. Now there are two types of active transport. There's primary active transport and there's secondary active transport. In primary active transport, what you have is all the same condition. The difference is that in primary active transport, the energy that is gotten, or that the energy that is used, is gotten from hydrolysis of ATP. So ATP is the source of energy in primary active transport. Again, ATP is the source of energy in primary active transport. An example of primary active transport is in sodium potassium pump. Check out our video on the sodium potassium pump. Moving on, we have secondary active transport. Secondary active transport uses energy, but the energy is not from ATP. The energy is from electrochemical gradient. Electrochemical gradient, or any energy currency aside ATP. Take note. That's the difference between primary active and secondary active transport. And secondary active transport carries more than one substance. Now, the different transporters that are used. Transport proteins can either be uniport, symport, or antiport. Uniport transporter or uniport carrier proteins only carry one thing. An example is the GLUT transporter. Glucose transporter only carry glucose. Are we good? Alright. Then you have symport. Symport are transport proteins or carrier proteins that carry two substances in the same direction. So an example is the sodium amino acid transporter, sodium glucose transporter, and other transporters. And the last one you have is the antiport. The antiport carrier proteins carry two things but in opposite direction. An example is sodium and calcium transport. Another example is sodium and potassium transport. One is going in, the other is going out. So those are carrier proteins from the uniport to the symport to the antiport. Then the third type of transport we have is vesicular transport. In vesicular transport, the substances that are moved are large. So we have it divided into some conditions. We have bulk transport. In bulk transport, you have endocytosis. Endocytosis means we are moving structures inside the cell. Cytosis, cell inside the cell. Now endocytosis is now divided into two. There's pinocytosis and phagocytosis. Pinocytosis means the structure, the substance you are moving is in liquid form, so it is regarded as cell drinking, while phagocytosis means that the substance you are moving in is in solid form, so it is regarded as cell eating. Moving on, those are the types of endocytosis, pino and phago. Then we have exocytosis. In exocytosis, the structure you are moving is out, so you are moving from inside to outside. And then you have transcytosis. Transcytosis is a combination of endo and exocytosis. You are moving structures in out or out in. That's what transcytosis is about. That's bulk transport. And the last type of vesicular transport you have is filtration. Filtration means movement is based on hydrostatic pressure. So substances that move from a region according to their pressures. So move from high to low pressure. And that is seen in the capillaries, it is seen in the glomeruli, in the kidney. Check. Next up, we'll talk about cell signaling and communication. Now, cell signaling and communication are the various ways by which cells communicate to each other. Now, these mechanisms include, number one, endocrine. Endocrine communication means cells are communicating and sending signals to each other via hormones. Paracrine communication and autocrine. Paracrine communication means that cells are communicating by diffusion via nearby cells, by diffusion. And autocrine means impulse or signals that are released by one cell and responded to by the same cell. That's autocrine communication. So these are the various mechanisms by which cells communicate via ligand cyclic adenosine monophosphate pathways. Check out our videos on all of these things. Next up, we'll talk about the cell cycle. The cell cycle is made up of 4 stages. We call it G1S and G2M. G1 is the growth and preparation stage, where the cell is being prepared for the S stage. The S stage means synthesis of DNA. So it is that stage where DNA is synthesized. G2 is growth and preparation again for M, mitosis. Remember that mitosis is a multiplication division, and mitosis has P-mat stage. P is prophase, M is metaphase, A is anaphase, T is telophase. Check out our videos on all of this on the cell cycle for more. Now, the clinical biochemistry of the cell cycle is in cancer. The clinical biochemistry of cell physiology includes: number 1, cancer as a result of altered metabolism via the Warburg effect. 2, we have diseases, metabolic diseases like diabetes mellitus. We have endocrine diseases and many other diseases. Now, that's how this concept should be understood: clear, precise, and exam ready. If this has helped you, go ahead, like the video, drop a comment, subscribe, and tell me what topic you want next. This is Dr. EBM, where you don't just study medicine, you master it.